Okay, I'll just back it up a little bit. Essentially, to say that the way we can study this process of synaptic plasticity or long-term potentiation is using model brain slices. When you activate synapses, so when there's a lot of activity that is happening, that can actually potentiate the strength of the synapse, and that's done via NMDA receptors. They're absolutely required for this process, and that's what the figure on the lower right is showing, is that if you block these receptors, you cannot get long-term potentiation of responses. And so the way you can think of this is an analogy that Graham likes to use, is that the more often you go to the gym, the more you strengthen your muscle. Same applies for synapses. The more active they are, the more that they will potentiate. So I'm going to skip that. We've already heard a little bit about some of the properties of NMDA receptors, and they are quite unusual because not only do they require glutamate to bind, but you also need to remove this magnesium blockade. And so they are unique, and they're absolutely critical for controlling the strength of the synapse. Synaptic plasticity is what we call it. And you can think of it as equivalent to learning or information storage. And the NMDA receptor will also effectively control how much communication is going across the synapse. So the NMDA receptor does need to be very tightly regulated. Oops. Let's put that. You can strengthen the synapses, and that was the process of long-term potentiation that I was just discussing. And there's also a counterpart to that, which is called long-term depression or weakening of the synapses. And normally, synapses operate within this range of strength. What can happen is that if synapses are not properly regulated, and that can be because of NMDA receptors not working properly or other gene products not working properly, is that if synapses are underactivated, for example, you can lose these synapses, and that can lead to disorders such as Alzheimer's disease and depression. The opposite can also happen when you overactivate synapses. That can lead to epilepsy or chronic pain. And one of the methods that, of course, is important to work on is finding new drugs and modulators that can control the NMDA receptor activity and put it back into this normal operating range. We did hear about memantine, which is being used for late-stage Alzheimer's disease, and also ketamine, which is being used as a antidepressant. So it's important to understand the way that NMDA receptors go wrong in disorders in order to properly target therapies. And now we're learning more about the detailed structure of the NMDA receptor and how variants can contribute to disease. This will be discussed in more detail by Dr. Furukawa. Thank you.
Thanks very much, John. So far, we've not had any questions in the chat, so let's carry straight on to Dr. Furukawa. Thanks.
Okay. Can you hear me? Okay. Sorry, I'm just gonna share screen now. Okay, so if you could see the screen, I'm gonna start my presentation. So thank you so much for having me in this GRIN conference. My name is Hiro Furukawa, and our mission in my lab is to visualize NMDA receptors at atomic resolution. What I mean by atoms, NMDA receptors are made out of amino acids, and amino acids are made out of atoms. So to visualize atoms, you can not only understand the shape of NMDA receptors, which looks like that in the arrow, okay, but you could also help develop therapeutic compounds. We could actually visualize compounds that are binding to NMDA receptors just like that. Again, this is all happening in the past 14 years in my professorship at Cold Spring Harbor Laboratory, which is right outside of New York City. As John beautifully introduced, brains are made out of neurons, as you know, billions of neurons, and then they basically exchange information at the specialized junction called synapses, where NMDA receptors encoded by GRIN genes are located. And NMDA receptors are shaped like that. This is the representation. It's actually the actual density, or what we call the raw data for NMDA receptors. So it's the image of NMDA receptors at high resolution, where we see gluon1 and gluon2b in magenta and green, dark green, and they are encoded by the GRIN1 gene and GRIN2b gene. Okay? And we also have what we call structures of GRIN2A or gluon2A as well. All right. For that to happen, so we call this methodology structural biology. So our aim is to, again, visualize NMDA receptors at high resolution. For that to happen, we have to figure out the way to mass produce NMDA receptors. And we're not using a natural source. We have GRIN genes in the form of DNA, and we incorporate DNA, for example, GRIN1 and GRIN2B DNA, we incorporate them into mammalian cell cultures instead of the brain, and let the mammalian cell culture produce NMDA receptors. And we extract and purify them, and then apply a technology recently called cryo microscopy or electron microscopy. So we're using an electron microscopy technique to visualize those. And then we need to have a way to interpret structures.
Yeah, sure. The first question then was about whether we can see anything around loss or gain of function manifesting directly in NMDA receptors. Does anyone want to have a go at that?
Yeah, so certainly we can record from we can isolate NMDA receptor mediated currents and establish whether you know, there's increased function. We can also run long-term potentiation assays to you know, as long as the receptors are there, we can establish whether they're contributing more or less to the assay that we measure in brain slices. So there are several ways.
But so the structure of the NMDA receptor itself is unchanged, but the function is different depending whether it's a gain or a loss of function variant affecting it. Would that be fair to say?
I suspect you could probably have both situations.
Okay, thank you.
So the bottom line, and I'm really sorry about the technical issues. I was supposed to have movies here, but the bottom line is that we can now mass produce NMDA receptors recombinantly. So it could be a wild type regular NMDA receptors or the mutated NMDA receptor variant. So we could just mass produce them and visualize them under microscope. The type of microscope we're talking about here is electron microscope. Okay. So what we end up doing is to basically just purify NMDA receptors. And if you could see the screen, this is the representation of NMDA receptor images under a microscope. So you can see black dots. So those are NMDA receptor protein molecules in various orientations. So what we end up doing is to pick them up one by one. So we typically collect about million, I'm going to call it particles, million NMDA receptor particles in random orientations, and average those, and you can start to see the features of NMDA receptors, which looks like that. And then now we can reconstruct, we call it reconstruction, but we can just take an of random orientation of NMDA receptor particles and reconstruct them into three-dimensional structures. And we can attain atomic resolution. So we can see atom by atom structures, which looks like that. And also the key advantage is that NMDA receptors takes different shapes. We call them conformations. And conformation movements are extremely important. And again, sorry for the technical difficulties. The movie isn't working now. But the bottom line is that we can actually see the pattern of NMDA receptor molecules moving when it's activating and when it's inhibiting, right? And the reason these structures are useful is because we can now model, oh, it's not working. Anyhow, so this is the structure of NMDA receptors. And what I wanted to show you in this movie slide was the location of green mutations. So we have the blueprint for NMDA receptors. Okay, now again, we can visualize them at the atomic resolution. What it means is that we can now locate where the particular green mutations are within the context of the structure. For example, the recent mutation that we've been working on, which is methionine 812 to leucine, is located here. So we can actually pinpoint where the residue of mutated site is. And what it allows us to do is to really figure out what it does in terms of NMDA receptor movement or basically function. So we can explain why this particular mutation is positively, we call it potentiation, upregulating NMDA receptor activity. And we're basically just hoping to do that for every single mutations that are available, that are happening. And then hopefully we can come up with a way to regulate the kind of abnormal way of moving NMDA receptors. The reason I'm saying that is that we can now also visualize compounds that are binding specifically to each individual domains. Like for example, if your mutation is, for example, located here, and if it's affecting the area called ATD, then there's also a compound that binds the amino terminal domain. So we might be able to regulate or fine-tune the activity of NMDA receptors. So again, my apology for the technical difficulty. I'm gonna try to find a way to upload the movie file later on. But the take-home message is that our mission is to visualize NMDA receptors at atomic resolution, and that would allow us to understand how green mutation is affecting the function of normal NMDA receptors. And also, we can see the compound or therapeutic compound, so that would also allow us to develop compound or novel compounds that specifically target the receptors that are problematic. So with that, I like to also, so this is my scientists at Cold Spring Harbor Lab, people who are actually tackling NMDA receptor questions. Some of the work that I presented now was done by some alumni members who basically just went out and doing the independent research. I wanted to also mention about the funding sources, especially Austin's Purpose, which is now a part of Cure Green Foundation. Austin is from Long Island, and this is the beautiful picture of Austin and his family, Paul and Awilda. Austin is now 17, and he has a Green 2A variant. And it's been an honor to be working with Austin for the past 6 years, I believe. And then just really happy that we're making some progress to understand his mutation and the structure and the collaborators. Thank you so much, and I'm happy to take any questions.
Thank you very much for that. I think the slide's got the end again, but we could certainly see most of the NMDA receptor cartoons. And that's fascinating to think how much depth we can get into now. And thank you and your team for all the work that you've done on this so far. Very exciting to imagine where we might go next. We've got just a couple of minutes left now before we're going to get cut off. And there's one more question in the chat from somebody who would like to ask a question specifically about their child who is 10 months old and has a gain of function with epilepsy. This question would like to ask whether there's any specific drug recommendations. And I appreciate that it's difficult to, you're not going to be offering any clinical advice, but are there sort of what groups of drugs would be best to treat gain of function variants?
So, I mean, if I could go, we definitely have to be careful, but if it's caused by activation, overactivation, if we know that this particular mutation is causing activation or overactivation, I think the best drug to, right now is to use memantine. There are many names for memantine. It's the basic name. Depends on who makes it. Some company sells it as Nemanda. It was originally developed for Alzheimer's reagent.
Okay, thank you very much.
And it is the blocker of NMDA receptors. So, yes.
Yes, I think there's going to be a lot more conversation at the next session after the break about gain of function and loss of function variants. So I'm sure anybody who's got related questions should log back into that one, which hopefully won't have the same gremlins as we've had, and be able to hear a lot more. I think that's us. We've come to time, so I think we have to say goodbye very shortly. But thank you both very much for your presentations today and for the work that you and your teams continue to do on behalf of all of our GRIN families around the world. Thank you very much.